So we've been talking about immune system, how immune system is able to make very, very diverse types of antibodies. These antibodies, we said, can bind specific antigens. Antigens are generally foreign in nature. So what good is it that when antibody binds an antigen, what good does that do to us, for us? So let's look at that, how the immune system is complemented by a different system altogether, helping it eliminate pathogens. Most of the time, binding of antibodies to antigens perform no useful function until and unless there is an effector mechanism that can cause an effect which would be destruction of the pathogen. Complement system serves that effector role. Complement system provides the actual protection from the response, while the interaction of antibody and antigen provides specificity of response. Think of it as a laser guided bomb. So our antibodies are going to go tag the hostile force, or it is going to point out where the enemy is, and the bomb is going to go and find and destroy that target. So targeting mechanism is controlled by antibodies, and explosive force is the complement system. That would be one way to think about the complement system. In other words, antibodies finger the target and complement system destroys it. The complement system consists of about 30 proteins circulating in blood plasma. These proteins are synthesized mainly in liver, and they are approximately 5% of the total globin fraction of the human blood serum. Most of these are inactive until they are cleaved by a protease, which in turn converts them into a protease. So to activate complement system, we basically need enzymatic activity, a protease activity. The protease will cleave these complement system proteins, which in turn are themselves proteases, and they will basically cause that effect. So now let's see how this effect begins. First of all, antibody will bind an antigen which could be present on the surface of a pathogen, a bacteria perhaps. This binding will cause a conformational change in the antibody, which will be read by a specific protein part of our complement system called C1 complex. This actually is a complex of 3 different proteins: Q protein, R protein, and S protein. So the C1 complex consists of 6 molecules of C1 complex consists of 6 molecules of C1Q, 2 molecules of C1R, and 2 molecules also of C1S. Either by C1Qs and 2 molecules of C1R and also 2 molecules of C1S. When the binding occurs between the antibody and the Q part, C1Q of this C1 complex, there's a conformational change, and this complex, basically this change is that now the C1S and R are now activated, and they are activated on the surface of a pathogen. Here is our complex, C1 complex, made up of Q molecules, R and S's. So when this happens, R and S molecules, they become active. These are proteolytic enzymes, and they will cause proteolysis of other substrate proteins. So when this is activated, C1, S and R are activated, they basically cleave a protein called C2 and C4, producing 2 fragments of C2 and C4. Here you can see this complex C1 complex has cleaved C proteins into C2A. Here's the C2A, and here's the C2B. C2A moves away and C2B stays there. It binds another protein which is also cleaved by R and S proteins called C4. C4 is cleaved into 2 proteins, C4A and C4b. C4b and C2b, they join each other and they decorate the surface of the pathogen. So this is the plasma membrane of the pathogen. The antibodies bound a specific antigen present on the surface of this pathogen. This binding caused a conformational change which allowed C1q to bind these antibodies, resulting in activation of C1r and C1s. These two molecules are themselves enzymes which cleaved one molecule of C into C2a, C2b, and also a molecule of C4, which was converted into two molecules of C4a and C4b. So this basically complex assembles on the surface of the pathogen, and C4b will bind the sugars present on the surface of the pathogen covalently and also will recruit the C2b. C2b, as you can see, is binding C4b, and this basically is the start of the process. Here I would like to mention that this is also called C3 convertase, because this is now going to cleave another protein called C3. And you guessed it, there are going to be two fragments generated by cleavage of C3, C3a and C3b. Okay, so C3a will diffuse into the surrounding, C3b will bind covalently to glycoproteins scattered across the cell surface, and macrophages and neutrophils have specific receptors for C3b, and they can bind C3b coated particles. And as you know, once that happens, this recognition takes place between the receptor present on the macrophage or neutrophil and C3b. It will trigger a receptor mediated endocytosis, and this particle, whatever entity it was, it will be basically engulfed by these phagocytic cells, and this is basically how this particular pathogen will be destroyed. Here I would also like to mention that C4b is an opsonin. Basically, it is preparing the bacteria for eating. So it is preparing the bacteria, that is why it is called an opsonin. C3a, which diffused in the surrounding, is a nephrotoxin. What is a nephrotoxin? It basically is going to result in release of histamine from the mast cells, increase the permeability of the surrounding tissue, as we know that will help facilitate movement of phagocytic cells in that vicinity. So our antibody targeted the pathogen, our complement system decorated, the process we said is also called opsonin, put decorations on our pathogen which will be recognized by phagocytic cells or eating cells. They will come and they can destroy this particular pathogen. So this is not the only way how complement system can help our immune system destroy the pathogens. There are other ways, we'll talk about them. Currently we were discussing the classical complement system pathway. We will see how this complement system, classical system, complement system pathway can also, another way, it can help our immune system destroy the particular pathogen.